In this video, we're going to review some basic principles of organic chemistry that relate to biological systems. One of the main properties that determines the behavior of a molecule is its polarity. Polar molecules are either charged, such as ions that have charge, or have charged regions. Water, for example, is a polar molecule. Although water is neutral, the higher affinity of oxygen for electrons compared to the affinity of hydrogen for electrons makes electrons spend slightly more time near the oxygen atom than near the hydrogen atom. This results in a partial negative charge around the oxygen atom and a partial positive charge around the hydrogen atoms. These partly positive areas of one water molecule can interact with the partly negative areas of another water molecule, and this is what we know as a hydrogen bond. Polar molecules can interact with each other, and non-polar molecules can interact between each other. However, polar molecules do not interact with non-polar molecules. Now let's look at the most common functional groups found in biomolecules. A functional group is a chemical group of molecules with specific chemical properties. For example, the hydroxyl group, which is composed of an oxygen and a hydrogen atom, is characteristic of alcohols and carbohydrates. The carbonyl group is present in aldehydes and also in carbohydrates. The carboxyl group is characteristic of organic acids, and it is found in proteins and in lipids. Proteins are made of amino acids, and the acid is provided by the carboxyl group. The amino is provided by the amino group. Therefore, the amino group is found in proteins and is also found in nucleic acids. Lastly, phosphate groups are part of nucleic acids too, and they're the ones joining the nucleotides together. But they're also important in short-term energy storage, as we're going to see in a few weeks. Molecules can exist as isomers. Structural isomers have the same chemical formula but have different structures. Glucose and fructose have the same chemical structure, as you can see here, each with 6 carbons, 12 hydrogens, and 6 oxygen atoms. However, the carbon skeleton is quite different. They differ in the position of the carbonyl group. Enantiomers have the same empirical formula and have the same carbon structure. Their difference is the orientation of the chemical groups around the chiral molecule. The chiral molecule results in 2 molecules having the same structure, but one being the mirror image of each other. And even though it seems that both orientations will be similar, they have big consequences for biological systems. For example, an enzyme can only recognize a specific isomer and it will not interact with its mirror image. It's like trying to give a handshake with your left hand. Now we will study the 4 groups of macromolecules that compose living systems. These molecules are usually classified as carbohydrates, nucleic acids, proteins, and lipids. Often, macromolecules are the result of linking together similar subunits, which we know as monomers, resulting in the formation of a polymer. Let's look at these 4 major types of biological macromolecules, and the polymers that form them, and the monomers, which are the basic units of the polymers. Important to note, lipids are not always composed of polymers the monomers linked together forming polymers. Carbohydrates, for example, are found in the chloroplast or in places of energy storage as starch granules or glycogen. Nucleic acids can be found in chromosomes. Proteins are found in intermediate filaments that give structure to cells, or they can also be found in muscle. Lipids, such as these fat droplets found in an adipose cell. The polymer of carbohydrates is, for example, starch or glycogen, and they're formed by linking together monomers, in this case monosaccharides. Their main function for energy storage, and they can also serve for structural support. Energy storage is, for example, in potatoes, or support such as cellulose in the cell wall of plant cells. The polymer of nucleic acids is, for example, a DNA strand or an RNA strand. The monomer is a single nucleotide that then is connected together to form a DNA strand. Their function in the case of DNA to encode genes, in the case of mRNA or RNA for gene expression, and you can find them in the chromosomes, or like we said, molecules such as messenger RNA. Proteins are formed by long chains of polypeptides. And these polypeptides are peptides put together, or amino acids joined together. Their function, the function of proteins, is very diverse. It could be from catalysis, such as enzymes, to transport, to movement, support. An example of catalysts are enzymes, transport hemoglobin that transports oxygen in your blood, for movement, myosin, and for support, keratin, such as the protein that composes your nails. Lipids, one example of a polymer in lipids is a triglyceride, which is three fatty acid chains joined together by a glycerol. In this case, the monomer will be the fatty acid. Their function, mostly energy storage. They can also have structural functions, such as the lipids found in the cell membrane, and some can act as chemical messengers, such as hormones. Examples of this: butter, oil, the phospholipid in the cell membrane, cholesterol, steroid hormones. The way you form large molecules or large polymers from monomers is by a dehydration reaction, in which you remove a water molecule and form a larger molecule by putting together the monomers. How you break down large molecules is by a hydrolysis reaction. In this case, you add a molecule of water into the polymer and then break it down into monomers.